Hello everybody, this is just a makeup video of the lecture on vision. So I'm going to start by defining the visual stimulus, which is what we see, and then I'll go through the visual pathway starting from the retina all the way up to mid-level vision, so there's just primary visual areas in the occipital cortex, and then I'll go through the higher order visual processes, including motion and color vision and face and object perception. So, what do we see? Let's look at a pretty nice visual stimulus, right? So what you see in this picture? That's a Mona Lisa picture. But we could take this from a bottom-up perspective, and so start by isolating its sensory components, such as brightness and color, and there's some contrast, and there's some various levels of spatial detail, and there's orientations of different lines, and there's some different textures, right? So those are just more bottom-up descriptors of the sensory components that are actually hitting your eye, and these are the components that have to combine, that your brain has to combine to create a perceptual experience. You could also take, alternatively, a top-down approach and start with the cognitive components. You could start with something about the meaning, or something that is a long-term understanding that you might have had just by being alive in the world and knowing maybe a little bit of art history, right? You can start by understanding the meaning of this famous smile, right? You can start maybe by knowing a little bit about the historical context, and these are things that are not necessarily tied to the bottom-up components of the sensory information that went into your input, but you're using these top-down knowledge and top-down information or cognitive components to integrate with the sensory data that's coming in, and this you'll see this as a theme throughout this unit, not on vision but also on audition and motor and so on. Much of cognitive neuroscience is about how the top-down meets the bottom-up. Now, so let's go through the visual pathway, right? So starting with the level of the retina, right? So we know that this is, of course, a ventral view of the brain, right? So we know that our visual information comes through our eyeballs into our retina, and then it goes through several way stations all the way until the primary visual cortex. Now, the textbook has its own fairly detailed diagram. I'm just going to show the parts of the diagram that I need you to know, right? So that starts with the level of the retina, of course, and the retina is sensitive to information both from the left visual field and the right visual field, right? And so this information from left and right visual field are actually, both visual fields hit both sides of both retinas, and then from the level of the retina, the information goes through the optic nerve, right? And we'll talk much more about how the retinal processing happens, but the information goes through the optic nerve, it crosses the optic chiasm, right, which is the white matter bundle that actually crosses over the midline as an important part of the visual pathway, right? So beyond the optic chiasm, that's when the left visual information from the left visual field and the right visual field actually gets separated, right? So the left information from the left visual field goes to your right visual cortex, and information from your right visual field goes to your left visual cortex, and that's because of the crossover at the level of the optic chiasm. Okay, so then past the level of the optic chiasm, we're on to the lateral geniculate nucleus, or LGN. And so this is an important way station that is still within the thalamus, which then sends its input into the visual cortex. And that is through the optic radiation, which is another white matter bundle. Right, so we have a combination of white matter areas, such as the optic chiasm and the optic radiation, and then we have gray matter areas, such as the lateral geniculate nucleus and the primary visual cortex, right? And all of these gray and white matters are coming together to form the primary part of the visual pathway. And among the early part of the visual pathway, most of these cells that are in the optic nerve and optic chiasm are together include retinal ganglion cells, which are a specific type of neurons that form a certain shape that we'll talk about in a bit. And then there's this important division that's between what I would call a majority pathway and a minority pathway. So the majority report, right, majority of the visual input goes through this lateral geniculate nucleus all the way to V1. Right, so that's the major pathway that I've just talked about. But then there's also this minority report, right? There's this minor pathway that goes through another area within the thalamus, known as the superior colliculus, and that actually then goes through the superior colliculus to the primary visual pathway. And so there is this distinction between the majority and minority pathway, and another important point to remember again is this crossover between the left and right visual fields and the left and right visual cortex. Right. So again, left visual field information, after the level of the optic chiasm, goes towards the right primary visual cortex, whereas information from the right visual field ends up in the left visual cortex. So there's this crossover that happens at the level of the brain. Now, and then another kind of corollary of this majority versus minority report difference that I'm talking about shows up in this very interesting situations where you could have a lesion in some parts of the pathway, right, such as in the optic radiation. And in some cases, this could give you hemianopsia, right, which is just the lack of vision in one half of the visual field, right? So if you don't have a right visual field, if you don't have a left visual cortex, you won't have information from the right visual field. So that's hemianopsia. And then there's also another interesting situation known as blind sight, which is when you actually have only an injury to the majority part of the visual pathway. So from the LGN, right, the lateral geniculate nucleus and its synapses towards the primary visual cortex. If you have a lesion in that pathway, but you nevertheless have an intact connections through your superior colliculus, right, which is another part of the thalamus. So your majority report is gone, but your minority report is still there. This could actually lead to another condition known as blind sight. So what's blind sight? Blind sight is actually a really interesting condition that kind of shakes how we think about awareness or consciousness. And so that is when people are blind, right? So these are people who have cortical blindness due to lesions in their visual cortex, right? So they say they can't see anything. They say that you put something in front of them and they say they can't see it. But if you throw a ball at them, they can catch it. Another example is if I give you, a large object versus a small object, and I ask you to reach for it, or you're going to scale the size of your grip to according to the size of the object that's in front of you. And it turns out that, and this is very classic visual neuropsychology work, it turns out that people with blind sight, or even though they're blind, they say they can't see what's in front of them, they still nevertheless scale the size of their grip according to the object that's in front of them. So that again shows you that there's some residual aspects of vision that's still there in these people with blind sight, even though they have injury or damage to the optic radiation, which is the pathway that goes from, the majority pathway that goes from LGN to the primary visual cortex. Even though they have that, they have an impairment in that pathway, the minority report pathway through the superior colliculus is still intact, and this leads to this interesting phenomenon of blind sight. Right. So going back to a little bit to the retina, let's talk about just the psychophysics part, right? So how the stimulus actually relates to the visual experience. Well, it turns out that at the level of the retina, right, just in the visual periphery, we have a range of light sensitivity, right? So we have our visible light, of course, ranges in the spectrum of luminance, and that can go from very dim, right, to such as starlight or even dimmer, to through indoor lighting, which is kind of middle, and then it goes through sunlight to very, very bright, and to like so bright that you could damage your eyes, right? So there's a range of luminance, which you could think of in log candelas per square meters. And then what's important here is that at the level of the retina, we have two major types of cells, right? And so one of those cells are rods, and the other cells are cones. And we'll show some distribution of these rods and cone cells and their receptors on the retina in a moment. But the important thing I'm trying to show here is that cones are sensitive to to a higher level of lighting overall than rods, right? So while rods are sensitive to low lighting situations, cones are sensitive to high lighting situations. But cones are more sensitive to color vision, right? So cones have good color vision, whereas rods have poor color vision, or no color vision actually. And so this, as I promised, is a distribution of the different kinds of retinal receptors, right, the rods and cones, on the surface of the retina, right? So here we have, from the level of the fovea. The fovea is just the point of the retina that you're actually fixated on, right? So if you're foveating at something, it means that you're staring at it, right? So we'll call that 0 degrees away from the fovea, because that is where the fovea is. And then, and right where, and that turns out to, the fovea turns out to be about 10 to 20 degrees away from the blind spot, which is where your optic nerve leaves the retina, right? So there's a small spot where you actually don't get direct retinal receptors. So that's where the blind spot, that's called the optic disc. So that occludes a small part of your visual degree, a small number of visual degrees. Now importantly, there's this trade-off between the distribution of rod receptors and cone receptors, right? So the cone receptors, which again are sensitive to color and are sensitive in bright lighting situations, right? They're exclusively in the fovea, right? So when you're foveating at something, that's why you get very good color vision at the point of foveation, because your fovea has exclusively cones and no rods. Now, in contrast, the distribution of rods is highest about 20 degrees or 10 to 20 degrees away from the fovea. So this trade-off between rods and cone cells means that you can have a much better visual acuity during a low lighting situation if you actually foveate about 10 to 20 degrees away from the object of interest. So think about a nice example is if you're looking at the night sky, right? If you're trying to see a cluster of stars that are quite dim, it turns out that if you don't look directly at it, but you look about 10 to 20 degrees away from it, that gives you better low light sensitivity, because that then gives advantage to your rods, because there's more distribution of rods at about 10 to 20 degrees away from foveation. And so that means that when you're looking 10 to 20 degrees away from the light source itself, you actually get more information from the light, right? So it's actually, you're better at looking at stars that if you don't stare directly at them. So, and just as a side note, 1 visual degree, right? When I keep saying 10 to 20 degrees, 1 visual degree is about the width of your thumb if you're extending your arm away from you, right? And just looking at your thumb, that's about 1 degree. Okay, so moving on to let's zoom into the layers of the retinal cells, right? So I've already talked about how you have rod cells and cone cells, and they trade off at the level of the retina. So beyond the level of the retina, we have a number of different cell types, right? And that includes retinal bipolar cells, and then those synapses those receive input from the rods and cone cells themselves, and then they send on the input to retinal ganglion cells. Right, and these retinal ganglion cells that make up a major part of the early visual pathway, they're divided into parvocellular ganglion cells, which are relatively small, and magnocellular ganglion cells, which are relatively large. So magno just means large, right? So that's how you can tell those apart. And so these layers of parvocellular retinal ganglion cells and magnocellular retinal ganglion cells are quite well preserved all the way through your visual pathway, right? So even when you get to the level of the lateral geniculate nucleus, which is in the thalamus, you get these nice distinctions between layers of parvocellular cells and magnocellular cells, right? So you can think of those as the M pathways and the P pathways. And to the first approximation, the M pathways, right, the magnocellular layers, give rise to or map onto changes, the visual system's ability to detect changes and movement or motion. Whereas the parvocellular layers, there's more of them, right? There are more parvocellular layers, but they're smaller cells. So these parvocellular layers, in contrast, map onto or correspond to the ability to detect spatial detail, including form of the visual information, as well as brightness and color. So later today, we'll talk about the where versus what pathways. And so just for now, think of the M pathway and the P pathway as corresponding to the where first and the what pathways, respectively. Okay, so that's at the level of the lateral geniculate nucleus. So how does it move on to the V1 areas of the brain? And this is where we introduce the really important concept of the receptive field, right, which is the region of the space that elicits a response from a given neuron. So, so at the level of the lateral geniculate nucleus, each cell can be thought of as being sensitive to a certain point. Right, and so stimulation at a certain point or light at a certain point can give rise to or can drive that neuron in the lateral geniculate nucleus. And interestingly, there are cells that are that respond to bright points, but then there are also cells that respond to dark points. Right? So you can think of the cells that respond to bright points as on-center and off-surround cells, right? So the center-surround distinction is, is what lateral geniculate nucleus cells are sensitive to, and then in contrast, the dark responsive cells are off-center or on-surround cells, right? So on-center, off-surround, off-center, on-surround. So receptive fields can be thought of as, at the level of the LGN, can be thought of as concentric circles of center and surround areas. Now, so then, so that's at the level of the lateral geniculate nucleus. Then at the level of the V1 or primary visual cortex, the properties, the responsive properties of the cells are actually now different, such that instead of being sensitive to certain points in space, the V1 is actually sensitive to lines and edges and different orientations of lines. So a proposed mechanism for how the LGN actually directly translates into V1 neural code is that you could have several LGN cells that are then sending input onto a single V1 cell, right? So a V1 cell, which is sensitive to lines, might just be collecting input from a collection of different on-center off-surround and off-center on-surround cells from the LGN, such that, therefore linking a few points together to make a certain orientation in an edge-like manner. So we're moving on now to some properties of the primary visual areas. Now, the discovery of the primary of neuronal receptive fields in the primary visual area is pretty much one of the most important discoveries in modern neuroscience. So these men are David Hubel and Thorsten Wiesel. So Hubel and Wiesel famously discovered neuronal receptive cell sensitivity in the primary visual cortex of the cat. And that has since then been replicated in many different species. And for this, they got a Nobel Prize in 1962. So we won't really go into this, but if you want to watch, there's a YouTube video that shows you how exactly they found it out. So the story goes that they originally didn't know that V1 was receptive to edges. They thought that it was receptive to different points in space, just like the LGN was. So they kept using different slides. So these are the old-fashioned kind of rotating slides in the slide machine, slide projector that they were using. And so they had this cat that was just kind of had its head mounted and was looking at the screen, and they were projecting all these different slides of different points in space, and the cells were just not responding. So the study was just really not working out very well for a long time, until they had this one breakthrough, which was when one of these slides was just slide was stuck in the slide projector. And so in these old-fashioned slide projectors, when a slide is stuck, it results in this edge on the screen, which is just the edge of the slide, and then the cell went crazy. So the cell that they were recording from just started firing, and that's when they had the aha moment, right? So that was the moment of insight that led them to eventually find out that the V1 cells were selective to orientation and to edges, not to points in space. So this is a diagram of that exact lore, right? So this is a representation of a bunch of different cells from the primary visual cortex, right? And so you could see how this orientation, right? So these here are different stimuli, right? So this bar, right, this edge moving in a certain direction, doesn't drive these cells, but when this bar is moving is starting in this orientation and moving in that direction, that drives the cell. Right? So this cell is sensitive to this orientation, and so different cells have different selectivity for orientation and direction, and then what's also important is that different columns of cells within the primary visual cortex are sensitive to a continuous distribution of directions, right? So this one group of neurons might be sensitive to this orientation. The next group of neurons are sensitive to a slightly different orientation, and then the next group of neurons is sensitive to a slightly more tilted version of that, and so on. So there's this contiguous organization of cells, of cell selectivity from cells within different columns that are organized next to each other. So there's this columnar organization, and then another aspect of important information that they found out in the primary visual cortex is also ocular dominance, right? Which is the idea that contiguous portions of the primary visual cortex are sensitive to input from the left and right eyes, right? So recall that we said that the visual pathway is getting information from left visual field and the right visual field, right? And so after the level of the optic chiasm, left visual field is only getting information, is only sending information to the right visual cortex and vice versa. However, the input from the left and right eyes, left and right retinas are actually interdigitated, right? So that you can have one column of your visual cortex being responsive to left ocular input, and the next column being sensitive to the right, and so on, right? So there's this this continuous organization of input from the left and right eyes, and also continuous organization of different directional sensitivity. Right, so this is just saying the same thing, right? So you can have information at the representation at the level of the primary visual cortex that are interdigitated between left and right eyes. And those are also sensitive to different levels of the magnocellular and parvocellular pathway from the LGN. And then another important aspect of visual organization is that it's also organized by space, right? Not just by directional sensitivity, but from the level of the primary visual cortex, you can essentially map, right? Map the level of eccentricity away from, or the number of degrees, visual degrees away from foveation. And that corresponds actually pretty well to concentric portions or concentric circular or organization at the level of the visual cortex, right? So what do I mean by that? Right? If you take information from the level of the fovea and you start to move away from the fovea, right? So start from 0 degree visual angle and move 5, 10, 15, 20 degrees away from the fovea, and then you keep giving visual stimulation at the left and right visual fields, and then you measure corresponding activity at the visual cortex. It turns out that there's a logical organization where the further you move away from the fovea, the further you see activity in the visual cortex. Right, so this is can be found using a technique known as retinotopic mapping, right, which is just the technique of relating the visual input directly to the neural output. Right, so that means that you're doing a retinotopic mapping study, you would be taking some visual stimulus and moving it very systematically from the fovea all the way out to the outside of the visual field, and then trying to record the corresponding brain activity. And the general finding is that the primary visual cortex, which is in the calcarine sulcus of the occipital lobe, right? So we know that the occipital lobe is generally where vision happens, but especially the V1 is embedded in or buried inside the calcarine sulcus, right, which is this quite prominent fold that's in the occipital lobe. So from the calcarine sulcus, you can see these concentric circular-like organization of the visual pathway, of the visual cortex, that maps on quite well to the number of degrees away from the foveation point in your visual field. And so another thing to note is that much like the left and right visual field are flipped in the brain, bottom and top visual field, top and bottom visual field are also flipped in your brain. So the areas that are dorsal of the calcarine sulcus are sensitive to visual input in the lower visual field, and vice versa. Another aspect of having the two visual fields and slightly different information into your visual cortex is that you end up with slightly different views that are seen by the two eyes, right? So when you're looking at an object that's close to you, your left and right eyes are seeing pretty different views of the same thing, compared to if the object is quite far away from you, and in which case the information that goes into your left and right eyes is still a little bit different, but they are much less different than if the object were very close to you, right? So this difference between what's seen by your left and right eye, it's known as retinal disparity, right? So that's just what's different, the disparity between what goes to your left retina and what goes to your right retina. And because of this difference, varies by how close something is to you. Our visual system actually uses retinal disparity as the depth cue, right? So that's how you can, people have designed some, fun toys like this one, right, where you can build in retinal disparity cue into something that's actually just two-dimensional, but the normal visual system would actually infer a three-dimensional experience from it, right? So this is one of those interesting tricks that you could play with the fact that our system is there's a top-down and a bottom-up um, information that our visual system is integrating. And so because we've gotten so good at taking the retinal disparity cue and inferring a three-dimensional structure and inferring that as a depth cue, people have actually taken advantage of that and just used retinal disparity to trick the brain into thinking that we're seeing a depth cue. And that also helps visual neuroscientists decide on certain experimental setups that actually make use of this difference between the left and right eyes. And so these are a whole set of studies known as binocular rivalry experiments. And so this is, for a long time, a lot of people are interested in, who are interested in the visual correlates of consciousness, or neural correlates of consciousness or conscious awareness, have been trying to use the setup to kind of get to it. And so so the way that it works is you present subtly different information to the left and right eyes, right? And so when you do that, the visual experience ends up flipping back and forth between what you see in the left eye and what you see in the right eye, right? So these are some examples of different visual stimuli that are coming in, right? And so this is one other set of possible visual stimuli, right? So if you get a left eye a stimulus like this, and a right eye stimulus like this, right? Horizontal and vertical, the percept when you're looking at both eyes, looking through both eyes, can either be flipping back and forth between what's seen in the left and right eye, or emerge between the left and right eye, right? So even though you're showing constant stimuli, you might result in a changing level of awareness. And so this allows neuroscientists to record or to look into the neural correlates of consciousness, which is distinct from the neural correlates of stimulus perception itself, right? Because again, the stimulus is the same, but our awareness or our experience can be flipping, right? So that's a really neat use of this binocular rivalry setup. Okay, so what else does our visual system use in deriving a visual experience from visual stimuli? So there are a few clues that you can use. So one piece of information is luminance, which is just how much light hits the object. Another piece of information is reflectance, which is how much light bounces off the object. And then a third piece of information is transmittance, which is how much light shines through the object. So by combining these different visual cues, we can infer the size and the material of the object. And again, so these are different kinds of cues that the visual system combines to infer information about the visual stimulus. And interestingly, you can also use top-down information to gain to make some inferences about things like shape and position of objects. Right? So in addition to luminance and reflectance and transmittance, you can actually have equal luminance situations, right? So where these two dots, for instance, they're the same in luminance, right? So the amount of light hitting your retina from this dot and from that dot are the same, which is the same as this. This is just a key, right? But because what's around those two dots are different, that could lead you to trick you into thinking that the two dots are actually different, right? So this is an example of context or top-down information actually changing how you infer the luminance cue. And this is another piece of information. Because of this queue of different of a three-dimensionality, right, in an object, you might think that um, this patch of within this image is lighter color than this patch within this image, but they're actually just the same, right? So again, this is a situation where top-down information, such as visual context, can trick you into thinking, into inferring different levels of luminance, even given the same actual luminance Q. So this is another of those interesting situations where top-down meets bottom-up. All right. And so it's an interesting illusion, right? The thing that we just saw, it's just by having a little bit more top-down information, you can trick the visual system into perceiving luminance differently. And this also applies to higher level visual processing, such as motion processing. So this is very clear in these geometric illusions, right? So there's a bunch more on this website that I've linked to, I've pointed to here, right? But I think most of you have probably seen some versions of these, right? So these here are two parallel lines. Right? They don't look parallel just because of these radiating lines that are coming out of them at the same time, right? So because our system is so used to seeing, when we see these radiating lines, inferring some kind of three-dimensional experience, you end up, kind of tricking yourself into thinking that these lines are not parallel, right? So same goes with the Ponzo illusion and the Mueller-Lyer illusion. Even though the two lines are always the same, because of the contextual cues, you end up thinking that, or you end up experiencing differences in length that are related to our three-dimensional our three-dimensional experience of the world, right? And so this is goes back to what we know as the inverse problem in optics, right? So we've talked a bit about the inverse problem before when we were talking about how we can infer three-dimensional data from a three-dimensional source from two-dimensional recordings. So this is in the context of EEG. When we're talking about EEG, we know that we can only record from the surface of the head, right? So we've got a two-dimensional array of electrodes that we're placing on the head, and from that two-dimensional array, we're supposed to infer a three-dimensional neural source, right? So we're somehow supposed to guess where in the brain activity is coming from, even though we're only recording from its surface. Right? So in vision, you get the same problem, right? You've got a two-dimensional experience. You've got a two-dimensional bunch of snapshots of the world, right? Which is what's on the retinal image, right? What is projected onto your retina. But somehow you're supposed to infer a three-dimensional experience from it. So how do we do that? Well, we use ecology, right? We use our experience with the world, and we bring that experience to bear. And this is all top-down knowledge, right? And so this is how we can use other kinds of information, such as contextual information and top-down information, to infer a three-dimensional experience from a two-dimensional image. And so because of that, this also relates to how we could see these illusions, right? So if you're living in a world that is carpentered, so where carpentry, of course, means that you're building things out of straight lines and beams and putting them together. So a carpentered world is a world in which you have lots of straight edges and right angles, right? So you have these 90-degree angles. And so the carpentered world hypothesis posits that we are experiencing these illusions just because we are used to a carpentered world. And so, this is in the 70s, there were actually a bunch of studies that looked into this carpentered world hypothesis by going to places where people don't tend to use straight lines as much, or experience where houses, for instance, aren't made of straight angles, and 90-degree angles, and straight lines, and actually measuring these people's perceptions from geometrical illusions. And it does turn out that there are some cultures that don't live in such a carpentered world that actually experience less of these geometrical illusions. So I think that's kind of cool. So another of these fun illusions. I'm not going to click on. Oh, so actually, yeah, this is the YouTube video where this came from, but I will show you this motion after effect. So you just have to follow the on-screen instructions and just stare at the thing until it changes, and then hopefully you'll experience the motion after effect, also known as waterfall illusion. All right, cool, right? So hopefully, if you actually stared at the screen the whole time, you would have experienced the motion after effects, which is the waterfall illusion. So even though the waterfall in this picture is not going anywhere, it's just a stable image. Um, what you saw, what you might have experienced after being adapted to the upward motion in the preceding screen, what you might be experiencing when you're given a static image now is the downward motion of the waterfall, right? So, again, this image of the waterfall is just a static image. There's no motion. The water is not moving in the waterfall, but it's called a waterfall illusion because after you've been adapted to upward movement, you now perceive downward movement even though it's not there. So this is another of those interesting examples of top-down meeting bottom-up, right? So you've got bottom-up information, you've got bottom-up information of motion, and then afterwards you've got no motion, but your top-down information kind of compensates for it by inferring downward motion even though it's not there. And so this is a nice way, these illusions are a nice way to test for areas of the brain that might be responsible for motion sensitivity, right? So even though you're not presenting any stimulus that has motion in it, you can measure the experience of the visual motion. So it turns out that our areas of the brain that are responsible for visual motion are are activated during the perception or during the perceptual experience of these motion after effects. And so this was done. These studies of motion after effects to study motion sensitivity in the brain was done by Theoret et al. A few years back, and what they did was give a moving pattern, right, that followed by a static pattern in which motion after effect was induced, and what they saw was that if you not only if you perform functional magnetic resonance imaging, you get a set of areas that are responsible for visual motion actually being active. Furthermore, when you use repeated TMS, right, when you take a magnet and you zap you stimulate the motion areas known as area MT or V5. So that's a hub of motion sensitivity processing in the visual system. When you zap this area MT or slash V5 compared to a few control areas, what you're seeing is that you experience less of this duration of the motion after effect, right? So suggesting that these areas are causally involved, areas MT and V5 are causally involved in the perception of motion. So this gets us to quite a general organizational principle of the visual system. After the level of the primary visual cortex, you actually get a distinction between a dorsal pathway that's responsible for motion sensitivity, also known as the aware pathway, and a ventral set of areas that are responsible for object sensitivity and face perception, and so that's known as the what pathway. Right. So the what pathway is ventral, and the where pathway is dorsal. And a few of the key players in the dorsal and ventral pathways are areas MT, right, which is responsible for visual motion, like we just said, and area V4, which is all the way towards the inferior temporal cortex, which is responsible for the what pathway. So object recognition in the visual system. And so this results in some interesting distinctions or dissociations between different kinds of brain damage and different problems with visual processing. Right? So if you have a damage in the area MT, which is part of your dorsal aware pathway, which goes dorsally all the way to the parietal lobe, what you would experience is motion blindness, also known as achinotopsia. Right? So motion blindness. So this is one example of a lesion in the dorsal pathway through the area MT. And so what people with achinotopsia experience, and there's a nice little YouTube link that you could click on if you're interested. What people with achinotopsia experience is the lack of integration of motion in the world. Right? So they're experiencing the world as if it's a series of static images, right? While most people are integrating different images to form a smooth perceptual experience of movement, right? Think of like a GIF, animated GIF, which is actually just a bunch of images that are stuck together. What we infer the movement, but people with achinotopsia don't infer movement, right? They just experience the world as static images. So instead of, for example, in this example of a person pouring water, instead of seeing a smooth integrated movement of water pouring and then the glass filling up, they're just seeing the water in the glass and then too much water in the glass. Right? So so that can make interacting with the world quite difficult. So in achinotopsia is motion blindness. Now, in contrast, you could have we also have the what pathway in contrast to the where pathway, and that includes areas after areas V1 and V2 goes towards area V4, which is important for color vision, and then towards the area inferior temporal cortex, which is in the temporal lobe. And so that's important for this. This ventral what pathway is important for color vision as well as for face and object perception. So let's just talk a little bit about color perception. Right? So we know that at the level of the cochlea and onwards, you have different kinds of cells that are responsive to different colors. Right? So within the cone cells, which are generally important for or sensitive to colors, you have some short, medium, and long cone cells that are responsive to shorter wavelengths, medium wavelengths, and longer wavelengths, which give rise to blue light and green light and red light, or orange to red light, respectively. So psychophysically, we can also think of different cues for our for color that include hue and saturation and brightness. So hue is the actual color itself, right? So that's the angular degrees around the cone that starts from red and going through orange and yellow and so on and all the way back to red. Saturation is the purity of the color, right? So that can be measured in percent from the center of the cone, which is 0, right, saturation, to all the way to 100% saturated. Right? So at 0% saturation, you basically don't have hue, right? Because white doesn't really have a color. And then we have brightness, of course, which is measured in percent, right? From black, which is 0% bright, to white, which is 100% bright. Right? So at 0% brightness, both hue and saturation are meaningless. So those are the different cues, right? Hue, saturation, and brightness are the different cues that you use to infer the color experience. But we also, again, have more top-down cues in addition to these bottom-up cues that we've just mentioned. So top-down cues include context, right? So and so by varying the context of a piece of an image, for instance, you can trick people into thinking that different patches of colors are different from what they actually are, right? So even though these two different cubes have very different levels of hue and saturation and brightness, you actually, because of the context, you actually experience the same overall colors, right? So this is, think of the dress illusion, right, which was all the rage a few years ago. That's another example of color contrast and color constancy, where top-down cues are playing tricks on how you experience bottom-up colors. And so this is one very nice example of retinal topic mapping meets color sensitivity, right? So these researchers were trying to find out how the visual system responds to color, specifically in the area phi4. And so what they were doing is trying to find the area phi4 by doing retinotopic mapping. Right? So their reasoning was that the visual cortex, an area of the visual cortex such as area phi4 should be sensitive to, should have a retinotopic map that is color sensitivity. That is color sensitive. So what they did was they took these rotating wedges of of like pie-like stimuli that were rotating at different at different times and that are constantly changing in color. And so when they do that, or they can kind of parcel out, they can find out which colors are sensitive, which areas of the brain are sensitive to which colors at a certain time. And what they found out eventually was that we have this concentric organization of color sensitivity within area phi4. And so we also have, when we have, there are some people who have lesions within area phi4, and that can lead to achromatopsia, which is a lack of color vision, which is different from just being red-green colorblind, right? So you can have, this is a pretty standard test for color blindness, right? So if anyone in the class is colorblind, right, they would not be able to tell, to read this, these numbers, right? But for most people, they can see that, this is the green color 74 on a red background, right? So if you're red-green colorblind, which just means that you're lacking the cone cells responsible for the red-green differentiation, right, then you would not be able to see these numbers. But cerebral achromatopsia is quite different from just color blindness, right? And so this actually is damage to the ventral occipital cortex, and that then affects color vision. So instead of seeing everything as just not having not red or green, they see everything as just different shades of gray. So let's final few minutes. I do want to talk a little bit about face perception and object perception, which is another major aspect of what the visual system accomplishes through its what pathway as opposed to the where pathway. And so one very important task that the visual system needs to accomplish is face perception, right? So, so for instance, if you need to recognize faces that are important or meaningful to you, and we know that there's some evidence for a face-sensitive area, which is in the inferior temporal cortex. So this is again part of the what pathway. But what Bob Desimone and Nancy Cowensher and a bunch of other people have done is record from human and monkey inferior temporal cortex and found that there are cells that are sensitive, that are highly responsive when they're given any kind of face, right? That is an intact face. So if it's a monkey looking at a monkey face, you get a lot of responsivity. If it's a monkey looking at another monkey's face, you also get lots of sensitivity. Even if it's a monkey looking at a human person's face, you still get lots of sensitivity. However, if you scramble the face, or if you occlude part of the face, or if you don't give a face, even if it's a biological object, right, part of the body, you don't get as much sensitivity in these cells. Now, there's another set of cells that are responsive for body rotation, right? So that are different from these face-sensitive cells. So these face-sensitive cells in the inferior temporal cortex, specifically, have become known as evidence for a face-sensitive area. And so this is known as the fusiform face area, right? And because that's part of the fusiform area is in the temporal lobe, and that it's part of the what pathway, and that has been known to be the seat of face recognition and face perception. And so damage to this fusiform face area can result in prosopagnosia, which is the lack of recognition of faces. And so this is a very, very good video, and I highly recommend that you watch it. I won't click on it right now, but you can click on it from the slides. And it shows this prosopagnosic patient, right, who can't recognize faces, even her own face. Right? And so it's a very moving demonstration of what it could be like not to recognize not only famous and familiar faces, but also oneself. But the upshot of that, I mean, the point I wanted to make with respect to the visual system is just that there's a face area that, there's a brain area in the fusiform gyrus, which is part of the inferior temporal lobe, which is part of the visual what pathway that is responsive to faces. And this whole story became much more complex and much more interesting, it turns out, when other researchers found out that it turns out that FFA is not uniquely face sensitive. It's also sensitive. It's also activated when truck drivers look at trucks, and it's also activated when bird watchers look at birds, and it's also activated when luthiers or music instrument makers look at musical instruments. And so it turns out that whenever somebody who's very well trained at something is looking at the domain or the set of things that they're very well trained in, right? So their trained domain of expertise, it turns out that you still get FFA activity. So some people actually thinking that the FFA is a domain general region as opposed to a domain specific region that is only sensitive to faces. FFA might be a domain general region that is sensitive to anything that you're especially an expert in. And very nice evidence for this view comes from the Greebles study. Greebles are just these made up little characters that are these three-dimensional, slightly anthropomorphic creatures that these researchers generated, and you could see how they're like all these three-dimensional little creatures, and they have these little features like little angles coming out of them, little horns coming out of them, and little limbs that are sticking out of the bottoms, right? And so what these researchers did was come up with a bunch of these three-dimensional creatures that were either in one group, in one family or another family. Right? So, and they trained people over the course of days to discriminate between these different greebles, and classify them into different families. And it turns out that after being trained on the on discrimination of or categorization of the different greebles, people showed FFA activity in response to the greebles. Right? So this ends up being very elegant evidence for the FFA as a pretty domain general region for learning and expertise. So that's all I have today. I think it would be nice if you have more time just to look through, to watch the video of the prosopagnosics, and also to watch the video of the Echinotopsia. That is here. Right. But I think the prosopagnosic video is really quite great. So I would highly recommend that you watch it. Okay, thanks.